In this course already, we've talked fairly extensively about DNA. We've talked about its structure, the phosphate sugar backbone connected to those nucleotide base pairs in the middle, which are all connected together in a hydrogen bond that form a double helix. We've talked about the process of replicating DNA so that your cell can undergo mitosis or meiosis. We've talked about how you can take DNA, unzip it, make little mRNA transcripts, and then later on turn those into protein. But I'd like to talk a little bit more about the numbers of just sheerly how big is how big is DNA. And I wanted to talk about how do we sequence it and how do we know what that genetic code says? How do we know what those base pairs actually say? I took a gene, I think this is cytochrome oxidase, and I just took part of it and I stuck it on a Word doc. If we go up to Tools and we go to, let's see here, we go Word Count. So this tells us about 2,272 letters fit on how I usually write. Arial, size 12, 1-inch margins. I also know for a fact that there are 3 billion base pairs in the human body. So let's go 3 billion divided by this number. And this will tell us that there are about, this looks like 1 million pages. So if you were to take your entire genome and write it down letter by letter, and you had a page that was set up like this, you would have over 1 million, 1.3 million pages of information. And all of that information is inside every single one of your cells. Every cell in your body has all of the DNA in it. That's a good test question, something to know. So every single cell has all of the DNA. Okay? And if you were to write down that code, it would be over 1 million pages long. Let's take a step back and talk about maybe a couple of things from the previous section, but let's talk about bacterial transformation really fast. So here's our plasmid, and don't worry, I'm going to circle back around to sequencing. We'll get there. Here's our plasmid here. Remember, that's a segment of DNA that a bacterium can pick up. And here is our target gene. This is human insulin, for instance. This is grown in bacteria, believe it or not. And what we want to do is take this gene and paste it onto our plasmid. So the way we do that is we digest each of them with a restriction enzyme. And so now we know what this little segment of DNA here and what this little segment of DNA here, we know that they're going to be complementary to one another. And so we can take this insulin gene and paste it into our plasmid with relatively high efficiency. And so we go ahead and, like I said, we always hijack things that biology has built for us. We don't actually design these things ourselves. So we took our restriction enzyme, digested both of these, and then we added our old friend DNA ligase that you should remember is going to repair sort of spaces between DNA segments. And now we have at the end our recombinant plasmid. But what if we're afraid the gene didn't get into the plasmid? Or what if we're worried that maybe some mutation has taken place or somehow something is wrong? Or what if we just want to study the plasmid to begin with? The way we do that is using electrophoresis. So, as I alluded to previously, what you can do is you can take and digest your plasmid and put it into one of these electrophoresis machines. Now, is DNA positively charged or is DNA negatively charged? Well, DNA is negatively charged, right? And so if you put a positive charge here, a positive charge here and a negative charge here, the negatively charged DNA is going to have to run through this electrophoresis machine. And as you can see here, smaller segments of DNA wind up running faster. And so what this is saying is there's a segment of DNA here that's 1,000 base pairs. And it's saying that there's a segment of DNA here that's 800 base pairs and one that's 200 base pairs. So what are we looking at here? What does this mean? And this marker, what you've put in here is just a known sample of DNA. So you know that this is 100 base pairs. You know this one's 200 base pairs. It lets you figure out what the other things are. But why is it that we have one that's 1,000 and one that's 800 and one that's 200? Well, what this is, this 800 base pair thing, is just the plasmid without the targeted gene. It's the plasmid without the gene that we want. And so you have an 800 base pair plasmid, and then you have a 200 base pair gene for insulin. So let's say we wanted to study this gene for insulin. We wanted to study this thing that we've inserted into the plasmid. What we're going to do is, and what they used to do in the old days is they would literally cut this band out of the gel because they knew that that's where their gene was. This is just theoretical, by the way. I don't know the actual size for the insulin gene. It's probably not 200 base pairs. That's not important. So let's say we have this segment of DNA here. This is our single-stranded segment. And we don't know that these are the letters yet, but we want to know what those letters actually are. So if we were to just go ahead and toss in our DNA primase, it would just replicate, right? It would just go ahead and replicate our segment of DNA, and if we ran this on a gel, we would discover that it's 14 base pairs long or whatever. And that's not super helpful to us. But what somebody figured out, and this is absolutely brilliant, that's why I'm telling you about this, is you can take and add a few T's that will stop the process. Okay, so this T right here does not allow elongation to continue. It stops that DNA primase right where it is. Or I'm sorry, I said primase. It's DNA polymerase. It stops that DNA polymerase right here. And so now if we run this on a gel, we have one band that's, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 base pairs long. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. And we are also going to have a band that is 1, 2, 3, 4, 5, 6, 7, 8, 9. It's going to be 11 base pairs long. Okay, so what's happening here is we just added in a very small amount of this defective T that stops elongation wherever it binds. So wherever you have one of these T's, elongation cannot continue. If we continue this process of making this DNA with our DNA polymerase and our defective T's, we are going to have segments of DNA that stop just like this. And so you have one that's sort of this long and one that's sort of this long and one that's sort of this long. Now if we run this on a gel, what are we going to see when we run this on a gel? Well, we're going to see the lengths of our DNA that contain this actual T. And I apologize, I actually flipped this, but I thought it would be easier for us to look at going from small to large. So here are our T's. We know that there's a T at the first position, at the 7th position, at the 11th position, and at the 14th. Okay, let's do the same thing now, but we're going to use faulty G's. And so again, these G's are going to sit down randomly as DNA polymerase is working its way across. Some of them are going to stop here, some of them are going to stop here, some of them are going to stop here, and you're just going to get sort of a random smattering of these different lengths of DNA. But what it tells you is the position of every single one of those G's. And you can use gel electrophoresis to figure out the location of the G's. We can repeat the same process, but using our C's. So now we know where both of the C's are. There could be more, but now we know where all the C's are. Finally, let's do this process. Let's do this process one last time, utilizing A's that stop the process. So again, what we're doing is we're adding these base pairs, and whenever they are incorporated, they will stop. And so if you do the math, if you do the chemistry correctly, you're going to wind up with relatively equal distributions of all of these lengths of DNA. So we've added our A's in, and we're going to run this on a gel. So, oops. Dang it. And so now we have our complete gel, and they actually do look something like this. And you can just read the positions of every single segment of this DNA. So the complement to our original segment was T, A, G, C, G, A, T, G, A, T, G, G, C. Or alternatively, you can think of it as, since we're reading the complement here, it was A, T, C, G, C, T, A, C, T, T, A, C, C, G. So this is a very old school way of doing this. And you would have to, you'd have to do this many thousands of times just to get one copy of one human genome. And so, again, this won a Nobel Prize because it is brilliant, but the techniques of modern sequencing are a little bit more advanced than this. But our class has been sort of very abstract and more like me telling you about stuff rather than actually showing you how it works. And so this is an old school way of sequencing genomes. Much of the Human Genome Project came together in this fashion using this kind of technology.